Hi folks, Kevin here. Well, today I've got a talk for you regarding the foundation or actually the footer of our soil talks. So I'm hoping to have multiple soil talks, composting talks, passive soil building techniques, more active thermophilic and vermicomposting techniques, and some of the ways we can speed up the breakdown of woody materials or cardboard and paper materials without so much labor force involved as well. But today's talk is going to build on the previous talk about sand, silt, and clay, and the previous talk about the hugelkultur pits. When I stripped all of that soil off the surface, all of that vegetative material, all of the topsoil, all the rocks and all, and buried them and covered it up with gravel, I just had dirt there. Yes, there were bacteria there and lots of weed seeds, because anytime the wind blows in, there's going to be weed seeds there. And what I wanted to do was speed up the succession, because I didn't want to grow lots of weeds. I wanted to grow vegetable crops. I wanted to grow perennial bushes, berry-producing bushes, nut-producing trees, fruit-producing trees. So that's further along in the succession, which is dependent on the resources that I put, the organic material that I put into the soil, and the microbes that are there in the soil, because it's the microbes that do all the work. So today we're going to do two parts of this. We're going to start off with one of my mind maps that goes through and introduces each topic and how we go through the successions, and then we're going to end with a great graphic that I found online. And that graphic, I don't know who to give credit to for it, because I look for a source to credit for it, but it really is the light bulb moment for me, showing how easily the relative number of organisms in soil samples, because if we know what's needed as far as microorganisms in each area of our plantings, whether it's, our food forest, or our vegetable garden, or our herb garden, or our perennial vines that we're growing out there, whatever it is that we're trying to grow, and if we want to suppress weeds, we know what we have to provide to that soil in order to optimize the microbes that are in the soil to get the desired results. And so this is going to go through the succession of the various plants and the soil components that are in the soil that allow that succession of plants to occur. So without any further ado, I think I said that right, without any further BS from me, let's get to the mind map presentation and then the diagram at the end. Thanks so much, and hang in there, folks. Okay, so the goal in this part of the presentation is to basically just give you a framework, a structure to it. I think it'll make more sense when I show the picture after this part of the talk. So it's a diagrammatic representation. Right now I'm just going to go over I'm going to try and lay the foundation for the image that's going to follow, basically. So when we're talking about the soil and we're talking about the big picture, we're talking about the succession of plants. We're talking about going from just plain dirt all the way to a mature forest, ultimately. So the nitrogen source, that's what's important. So nitrates predominate in dirt because it's all bacteria there. However, in a fully developed woodlot and forest, the ammonium levels are the primary nitrogen source. So we have less nitrates and more ammonium in a very mature forest, and we have almost all nitrates and very little ammonium in our bare dirt or in our weed lot. So we're talking about the succession of plants. As plants come in, as the stages of the development of plant growth change over time, going from bare dirt, creating soil, building more soil structure as we progress along the timeline. And this will make more sense as we show the picture later on. So we're going from weeds to woodlots. We're going from bacterial dominated to fungal dominated. We're going from primary consumers, which are decomposers, through increasing the number of secondary and tertiary and quaternary trophic levels. And that has to do with the soil food web, which is a little bit too much to add into this talk. I already tried adding that in, and I had to remove it so that this talk wouldn't be too long. So with bare dirt, basically it's just bacteria being present. So Weeds require high nitrate levels and very low ammonium levels. So whenever we plow or we till the soil, we're destroying structure, we're disrupting the fungi, the protozoa, the microarthropods, and the nematodes. And so what we're doing is we're setting the stage for bacterial-dominated soil and therefore high nitrate levels. So every time we till, not only do we bring the weed seeds to the surface by destroying the structure and inhibiting the growth of the organisms that depend on the fungi, we are therefore switching the balance back to a more primal successional level, primary successional levels, and creating more bacteria and therefore higher nitrate levels. So that's really creating a weed paradise. Very few fungi and protozoa are present in these soils that are dominated by weeds. What about our vegetable gardens? How do we go ahead and create a better soil environment for our vegetable gardens? So when we're talking about our vegetable gardens, we're really talking about early and mid-successional soil structure. So we'll go through that now. So early successional plants, the goal is to decrease weed production and increase our vegetable production. Most vegetables require a balance between the nitrates and ammonium levels. Now, when I say a balance, I'm not talking about an equal balance here. I'm talking about there's some ammonium detected in our soluble samples, but the nitrates certainly dominate. So there's many more bacteria relative to the number of fungi present in our early successional plants. So, but we do need more fungi as opposed to what we see in our weed beds. So by increasing our brown materials, we're increasing the amount of fungi, and therefore we're pushing our successions to a more mature, later stages of our successional growth, which will make more sense when I show the graph at the end of this. So here's an example of some of our cruciferous vegetables that really do like mostly bacteria, but they need some fungi as well. So we need more nitrates to feed these plants. They prefer higher levels of nitrates than ammonium, but they still need the ammonium being present. There's also protozoa detectable in these samples. So we're going to see high levels of bacteria, some fungi, and some detectable levels of protozoa there as well. So what about the mid-successional plants? Well, we're talking about the majority of our perennial vegetables, our herbaceous species, some of our root crops, some of our vining species. We're seeing an increased level of fungi present. We're seeing more protozoa detected, and we're starting to see bacterial and fungal feeding nematodes present in the soil. So now we've created an environment where there's consumers of the bacteria and the fungi that are present in our previous samples, and consumers of the protozoa as well. Well, not yet. Be just a moment. So now, let's say we're planting our row crops, and we want to create grasslands to feed livestock. Well, we need higher proportion of ammonium in this level. So as we're going along the scale, we're increasing the amount of fungi that's there and decreasing the amount of bacteria. Therefore, we have lower levels of nitrates. So our perennial grasses have higher protein content. Bacteria and fungi levels are about equal in number at this point. We have protozoa present in this soil samples. We have an increased number of both fungal and bacteria-feeding nematodes, and we have some predatory nematodes starting to show up in this level. So we're at about the mid-scale in our whole successional series, which will show up better in the graphic to come. What about our perennial shrubs? So as permaculturists, we want lots of various berry bushes and all. And all of our bushy and flowering species, nectaring producing. So now we're getting much more biodiversity. These are the plants that we see on the margins of on the edges of the woodlots as well.
So they need even higher proportion of ammonium levels relative to nitrate levels. So we're having higher numbers of fungi. So relatively fewer bacteria present in our soil samples, and we see predominantly fungi there. We see more protozoa when we look at a soil sample. We see more fungal and bacteria consuming nematodes as well. And now we're starting to see some predatory nematodes in the soil samples as we get into our more perennial hardy berry producing bushes and flowers. We're seeing microarthropods in our soil samples, and we need to use lots of our brown materials. And I'll go over our greens and our browns in some of the composting talks later on. Okay, now we're going to get to our forests and woodlots. So at a lower level, it would be the deciduous trees that we're having, our chestnuts, our walnuts, our pecans, our almonds, our maples, all of these various deciduous trees that drop their leaves in the fall and winter, and our oaks as well. So we have all of these trees, and then we get to an absolute mature forest. So as we progress through these latter successional stages, it's almost entirely fungal dominated. Vastly outnumber our bacterial population in these soil samples. Fewer protozoa, because the protozoa are feeding on those bacteria. We see stable numbers of bacteria and fungal feeding nematodes. We see more predatory nematodes in these samples, and we're seeing many more microarthropods as well. So now we're going to go over to a diagrammatic representation, a picture that I found on the web that I think really shows the scale much better, but at least you have this information you can go back to and look at to help you put everything in perspective and see the samples as well. Okay, so now we're going to talk about the succession of plants as we go from bare soil, such as an example would be in our last hugelkultur hugel pits that I talked about in a previous video. So that bare earth, which we're going to call it dirt, is over here on our left-hand side of the screen. And as you can see, it's all bacteria. There's lots and lots of bacteria in the dirt. Now we're going to start creating the soil food web as we progress along the right side of this image. So we're going to end up in old growth forests, but we're going to gradually get to there. And when we're talking about this, we're talking about basically we're going from greens material to browns material if we're talking about composting, and I'm talking about relative proportions. We're going from high nitrate levels to high ammonium levels, going from left to right on this scale as well. On the left-hand side, we start off with basically a moonscape, no organic material, and as the organic material falls to the surface of the soil, then bacteria are present and they start to decompose it. So remember, our bacteria and our fungi are both primary consumers, and they're decomposers. At least the fungi that I'm talking about here and the bacteria that I'm talking about in this particular talk. So the bacteria, when they decompose, their end product is high levels of nitrates. So we dominate when we have a weedy beds with nitrates, very, very high nitrate levels. There's very few fungi. So down here in this diagram, that little comma-looking white comma there is fungi. This little red dot is some protozoa. Now, protozoa consume the bacteria that are here. So again, weeds love lots of nitrates. What produces the nitrates? Bacterial-dominated soil. Now, when we get into our vegetable gardens, we're talking about early successional plants to mid-successional plants. And so some of our grasses occupy both of these areas, but our cruciferous and brassicas are all in this early successional plants. So we have lots of bacteria, but we're getting more fungi and more protozoa in this soil type. a good diagrammatic representation. Then when we start producing our eggplants, our carrots, our vines, our perennial herbs and all, when we're getting into our mid-successional plants, then we're reducing the relative number of bacteria. And you can see the scale of things. The bacteria population decreases as we go to the right, and our fungal population is increasing. Well, one of the ways the bacterial population is decreasing is we have more secondary consumers. We have the protozoa here consuming them. And now we're starting to get, when we get into our mid-successional vegetables and foods that we're consuming and our herbs, then we're starting to get some bacterial and fungal feeding nematodes. So when we do a soil sample now, we're seeing fewer bacteria, more fungi, some protozoa, and bacterial and fungal feeding nematodes. Now we're getting into our row crops and all, our corns, our wheat, all of our grasslands, the higher protein levels that we want to feed to animals, or we talk about feeding to the population as well. In this case, we have, again, fewer bacteria, but much more fungi. And if you look, they're relatively equal, the number of fungi to bacteria now. So we have equal amounts of both nitrates and ammonium levels here. So when we're considering what plants we're going to grow, what are our ideal plants that we're trying to propagate on our property, we try to look at, well, what group of plants do we want to grow, and therefore, how do we feed the soil to get the appropriate amount of organisms? I'm trying not to be confusing here, but as you can see, the bacterial population decreases, the fungal population increases, and then we get secondary, tertiary, and quaternary consumers along the way. So again, back to our field crops, our row crops, our grasslands that feed the herbivorous consuming animals that we care for as well. We're going to have equal numbers of fungi to bacteria. We have a stable number of protozoa. We have a more bacterial and fungal producing, consuming rather, nematodes. And now we're starting to get some predatory nematodes as well, and some microarthropods once we get to the row crops and all. Then we get to our berry producing plants, all of our perennial bushes and vines. Again, fewer nitrate producing bacteria, more ammonium producing fungal elements, more protozoa, more bacterial and fungal consuming nematodes, and far more predatory nematodes, and still some more microarthropods in these areas. So again, we're going from green feeding materials to more brown feeding materials to get these populations in the right numbers. Then we get to our deciduous trees and our old growth forests and all. And basically, we're just gradually decreasing the number of bacteria in the soil samples, increasing the number of fungi that are there. And ultimately, we start to, as we get more and more mature, since there's fewer and fewer bacteria present, more and more fungal dominated, we start to reduce our numbers of bacterial feeding protozoa as well. And also, we start to stabilize the number of bacterial and feeding nematodes, but actually increase the number of fungal feeding nematodes, and we increase the number of our predatory nematodes, and certainly there's more microarthropods. Now here I'm just talking about the microscopic samples. So just to review one more time, as we're going from left to right, we're talking about high nitrate levels on our left side of the screen, high ammonium levels on the right side of our screen. We're talking about feeding the soil more green materials on the left-hand side of our screen to more brown materials on our left side, on our right side of the screen. Excuse me for saying that. So I hope this helps homesteaders, beginning gardeners, beginning permaculturists to see how we can look at our crops that we want to be considering planting, what our problems are. If we're having many, many weeds present in our gardens, then we can assume that there's going to be more bacteria there, and we need to get more browns in there. We need to get more of the fungi. And I'll go over some of the ways that we can relatively passively create more fungal-dominated soils by using paper products, cardboard products, wood chips, and so on, and actually getting some of the, I'm going to call it fungal duff from the wood lots. So, and they're around us, most of the places in the Northeast anyways. We can get very, very tiny samples of the base of the O horizon, and I don't want to go into that too much. And we can inoculate these materials, get them started, and then we can either compost them or even use them in mulching materials as well to help with our more passive feeding of our soil, changing the successional level more rapidly than nature would do it itself.
Well, I'm back. I hope that that was somewhat enlightening, helped to put things in perspective as far as bacteria, fungi, protozoa, the nematodes, the various types of nematodes, and certainly the microarthropods. And we didn't get into the insects and earthworms and all that yet, but at least seeing going from nitrate predominant to ammonium predominant, depending on what you're trying to grow and what you're trying to inhibit. You're trying to reduce the occurrence of the weeds. So I hope that was helpful. If it was, please give me a thumbs up, share it with your friends. Please interact with me. Let me know what you think. Are there any other resources that you think are good and that may help me to actually present this information or additional information in the future? I've got a whole bunch of ideas on soil composting and techniques I'm using around here, but I'm excited to learn. And I think you are too if you're at this point of the video. Well, thanks so much, folks, for hanging in there. Remember, leave a comment, subscribe, and pass this on to your friends. Take care and have a great day. Bye-bye now.